Hello, my name is Bradley Alicea, and I'll be presenting today on the interdisciplinary underpinnings of the neurosimulation of spatial intelligence. This work is in conjunction with the Orthogonal Research and Education Lab, and particularly our cybernetic interest group. So an overview of where we'll be going today. I'm going to sketch out a route to the neurosimulation of spatial intelligence. So we haven't done anything in detail, but this is just a route to getting there. The first, and a lot of these different topics are kind of related, and so I have this nested diagram. The first thing we'll talk about is vertebrate neuroanatomy, so thinking about the origins and correlates of spatial cognition in vertebrates. The next is ecological psychology and biocybernetics, so thinking about the brain as this sort of ecologically influenced structure, but also thinking about it as a circuit in terms of how biocyberneticists would think about the brain. The next is path integration and taxis, so we'll talk a little bit about path integration or route finding, and then taxis, which are movements towards different stimuli and movements along gradients and how those generate intelligent behavior. The fourth area is neuroethology, so we'll talk a little bit about neuroethology and how the brain interacts with sensory modalities to produce spatial cognition. And then finally, we'll talk about embodiment in GSLAM. So embodiment, of course, is very important for any sort of model of intelligence, as well as this notion of GSLAM, which engineers use to build control systems that are spatially embedded in embodied models. So when we talk about neurosimulation, we're talking about simulating the conditions of behavioral outputs. And so the neuro component refers to development, embodiment, sensory world, amplification, and evolution. Development often is associated with generativity, amplification with something called good regulation, and evolution is roughly corresponding to shared derived traits. The simulation part then replicates components of a system under plausible conditions. So to take all the aspects of the neuro and then simulate it in different ways. So developmental neurosimulation, which is something our group has worked on, is simulating the construction and expansion of the embodied nervous system. It allows us to ask what could happen in terms of morphology and behavior. From our work, we might aim to model something like simple connectomes, self-assembled, and an embodied context. So our first stop is to consider the vertebrate neuroanatomical view. So spatial navigation in vertebrates is typically thought of as a learning, memory, and representational phenomena based on a process of consolidating episodic memory. And so in mammals specifically, but in other vertebrates as well, spatially explicit behaviors have been tied to place and grid cells in the entorhinal cortex, which is mammal specific, and they're analogous structures in other vertebrates, but which provide a geometric representation or an abstract map of allocentric space, that space outside of the body frame. So if you go back to 2014, the Nobel Prize in Physiology and Medicine was awarded for the brain's navigational place and grid cell system. And this was awarded to two groups, one headed by Dr. John M. O'Keefe, and the other headed by Drs. Maybrit and Edvard I. Moser, for their discoveries of nerve cells in the brain that enable a sense of place and navigation. So you see the difference between place cells and grid cells. Grid cells set up sort of a coordinate system where you can have these periodic episodes take place, and then place cells represent specific discrete objects in the environment. These are the two papers that we'll be talking about here. The Hippocampus as a Cognitive Map, which is actually a book, and then the Moser's grid cell and cortical representation. So the cognitive map is a system which generates an absolute reference of Euclidean space and is located in the hippocampus. This is from John O'Keefe's work. And so it's kind of thinking about spatial cognition being located in a cognitive map. This approach was based on years of neuroanatomical investigation. So before the late 70s, there were a lot of lesion studies, behavioral experiments, and neuroimaging that had been done that had basically been confirmed by O'Keefe's team's work. So this work hypothesized place and misplaced cells. So place cells are information about the environment, and misplaced cells are displacement and exploration of the environment. So you have two different cell types, and they're working together in the same network to predict spatial locations and drive spatial behavior. Now, of course, we can look across vertebrates to see how diverse the hippocampus is. So the hippocampus is actually a homologous structure across vertebrates, and you see this in humans, of course, which is in D down at the upper right-hand side of the slide. You also see it in opossums, and that's, of course, also a mammal. Then tegu lizards and petromyzon, which is a fish, and you see those. They look a little bit different than the hippocampus that you see in the human opossum clade, but they still have sort of an analogous function. And so we can predict that there's a basal capability for hippocampal spatial processing, and this is like sort of the acquisition of it in the last common ancestor that had a hippocampus. And so we'll talk about two more papers. So the reason I showed you the phylogeny is because we're going to be talking about the zebrafish telencephalon, and then talking about maze studies, which you can investigate from rodents, which is this paper that's cited here, to fishes. One thing we can learn from the zebrafish larva is that place cell analogs are found in the fish telencephalon, and this is showing how place cells come online in larval development. Place cells in the fish telencephalon are enriched in telencephalic development, which happens during this larval period, and it forms a spatial population code in the adult. So here we have these place cells that are sort of forming in the early, we see like learning in the telencephalon during this larval development, and they go on to form the spatial population code, which we see. We also have these maze learning paradigms, so one of the things we want to think about is we want to think about embodiment and how organisms with bodies kind of explore their space. So it's not just with the brain, but space. And so there are these maze learning paradigms where learning utilizes place information and builds a representation of space. And we'll talk later about how space itself is represented through sensory information. But then we have to think about how spatial navigation and learning is acquired, and is it generally learned this way. So we might actually use these maze learning paradigms to train an agent or to train an animal, and that gives us interesting results. But in a naturalistic context, these mazes may be just kind of an artificial thing, and so the learning that we acquire might be an artifact of that. So what do you do if you do not have a hippocampus? So a lot of non-vertebrate animals or single cells conduct spatial navigation, but they don't have a hippocampus, and they don't have that kind of a learning structure. So what other anatomical mechanisms and functions are involved in spatial navigation? If we're interested in the simulation part, and even in the neuro part, we want to know if there are other mechanisms that are plausible. So this brings us to the ecological psychology and biocybernetics view. And in this part of the talk, we're going to look at this paper, Common and Specific Activation Supporting Optic Flow Processing and Navigation in Neuroimaging Studies. And so we find that functional segregation between optic flow processing, which is in the dorsal pathway, which sometimes is called the what visual pathway, is separate from spatial navigation, which occurs in the ventral visual pathway or the where pathway. So we have optic flow processing and spatial navigation happening in the visual system, and they're basically, the visual system is providing information of those two types of information to the hippocampus or to other spatial processing centers. And so one thing from the optic flow literature that's interesting is this idea of flow parsing mechanisms. And flow parsing mechanisms have been identified that extract object motion from middle temporal units in primates. So there's this spatial processing mechanism that is used to extract object motion from visual information, and this allows us to parse naturalistic flows. Spatially tuned units identify both object borders and relative or egocentric motion. So you have at least two mechanisms within the visual system that allow us to sort of parse information relevant to spatial cognition. And then, of course, we also have optic flow and allocentric navigation. And this is also another thing that happens to visual information to extract the spatially explicit information that can drive spatial navigation. And so if we look at a primate-centric model, remember this is just in primates, we have different types of cells. Now we have our place cells, of course, but we also have our head direction cells, and we have a representation of speed, so we have our place cells, of course, then we have head direction cells and speed cells, and those form this sort of network. And then you have vestibular and optic flow information. So the optic flow and vestibular information is integrated by head direction cells. So you see those two inputs to head direction cells. And head direction and speed cells iteratively update place cells. So place cells sit at the top of this hierarchy. Head direction and speed feed into it, these cells, their states, and determine the state of the place cell. And then vestibular and optic flow information are going into head direction, which completes the network. And so we're getting sensory information, and ultimately it's driving the behavior of place cells. If we think a little bit about these head direction cells, they're very interesting, and so maybe we can follow more up on that. So we have two papers here, a bio-inspired 3D neural compass based on head direction cells. This is an engineering take on this, where we're looking at this model of bat head direction cells, and then this more biological paper where they look at three-dimensional head direction coding in the bat brain. And it's based on this model of the bat. So the bat uses echolocation and vision to sort of tune their spatial navigation. They have this flight room set up where the bat is flying around the room. They're taking pictures of the bat, and they're looking at their brain. They're looking at the HD cells they're tuning with and their firing rate, and they're looking at it for different behaviors. They're looking at the tuning width in percent, and then they're looking at the peak firing rate in hertz. So this shows that there's this responsive head direction cells for different types of behaviors, and in flight there are different types of neural responses. And so if we put this into a phylogenetic perspective, we have primates, Chiroptera, and rodents. These are all mammals, but they're, of course, have different ways in which they treat optic flow. So in primates, we have optic flow contributing to a view representation. And this results in visual specializations in primates, but it has this view. This is why I showed the image of that network with the place cells and the visual information. So that's in primates. But in Chiroptera, optic flow contributes to a view representation as well, but this is due to visual and auditory specialization. So they have this view representation in Chiroptera is a sister outgroup of primates, so that makes sense, but they have this extra set of specializations. And then rodents, being kind of distant from Chiroptera and primates, optic flow contributes to a place representation where somatosensory and olfactory specializations dominate. And so you can see that there's this relationship between how information like optic flow goes into the brain, and depending on how the brain is adapted to different types of environments, different types of lifestyles, spatial cognition can occur in different ways, but kind of rely on the same substrate. And so this is just a little, on the lower right, there's just this little visualization of how in flies, visuomotor transformations basically stabilize flight through this optic flow mechanism. So this is actually quite a bit different than the mammalian last common ancestor, and this is quite different than the last common ancestor for mammalians, but you see that flies are also using optic flow to stabilize their movements, and then that translates into how they can navigate through space. O'Keefe and Nadel actually had a lot to say about Gibson, and the quote is, "Spatial cognition is a set of transformations acting on sensory input." Invariants, in the form of things like ambient optic arrays, are extracted from multiple sets of alternative viewpoints or experiences. And so this paper below on the ambient optic array and eye perception kind of shows what this looks like. So basically, you have all of these possible sensory transformations from the sensory world. We have this physical set of transformations and this visual set of transformations. And spatial cognition is basically acting upon these transformations, finding specific pathways forward. So you have all the sensory information, which gives you a lot of possible places to explore. Brain and spatial cognition filter out and leave you with a small set of alternatives that you can then explore. So sensory transformations can disambiguate objective or allocentric and subjective egocentric movement. These trajectories are the consequence of the sensory environment, but also the way the agent moves through the sensory environment, and it can kind of put things into different frames. And so, of course, then observers, or agents being observers, move to different locations relative to their target. And they have to be able to track that target as they move, even though the perspective changes and the object changes its kind of size and shape in the allocentric space. So this little set of pipes here is, if you have this agent here and it's moving in sort of an arc, it's going to take different viewpoints of this set of pipes, and it's going to look at it in different ways, and it's going to get different information, but it can always tell that it's looking at an arc. Furthermore, it can remember all those positions, and it can use those to navigate through these pipes, knowing relative information, relative allocentric information about the object in the environment. And so this paper by Rushton and Warren is moving observers, relative retinal motion, and the detection of object movement. Talks a little bit about this in terms of your agent moving over time at a certain angle, and then having these points of reference where their frame of reference shifts, and then they have to use their memory system to sort of align everything later when they go back to navigate it. And of course, we can look at very small connectomes to think about different types of plausible visual pathways. So what I showed you in the last slide was this image of all these plausible visual pathways where the brain filters out all of the implausible ones and leaves us with the most likely visual pathways. But in an anoid connectome, which is very small, it's less than 100 neurons, we can do these experiments where we can look at all the plausible visual pathways available to the anoid. And so we can look at this in terms of a 2-pixel system, which is the sensory motor photoreceptor that can spatially resolve light stimuli. And then the information is sent to the effector on each side, and it drives behavior. And then over time, you get these different nerves that are ipsilateral, and they cross the midline. And then you have these more evolved eyes, with lenses and eye spots and things like that. And then, so you build up from that simple model to these more complex models. And you can get these more complex behaviors like mutual inhibition, bilateral divergence, signal delays, and all of this. And so the reason I'm showing you this is to show you that spatial perception and spatial cognition can be relatively easy. You don't need to have a representational system. You don't even need to have that many cells. But this is also a hypothesized precursor to visual systems. So if this looks familiar, this is because it is the sort of biocybernetic approach, and it's similar to Breitenberg's approach to biocybernetics with vehicles and building vehicles that approximate small connectomes, nervous systems, things like that. We can also look at spatially distributed light sources, which require sensory arrays, internal pathways, and viewpoints on the light sources. So now you get an embodiment involved, and body location and posture become important. And we actually had an interesting discussion in one of our Saturday morning Neurosim meetings on Breitenberg's neuroanatomy and synthetic biology work. Breitenberg actually did a lot of neuroanatomy before he proposed the vehicles work, and he built these, he investigated fly connectomes quite extensively, looking at their neuroanatomy, looking at their output behavior, And this is kind of what led him to the vehicles model. So, there's a lot of kind of uh, space between fly connectomes and Bratenberg vehicles. It's really interesting biocybernetics, and I think has a lot of relevance to this topic. So go back to our July 19th, 2025 meeting for more. And we have this new paper out called A Brief History of Cybernetic Imperatives. This is on the PsyArchive. This is thinking about how we model plausible movements. So far we've talked a lot about vision and visual information, and then spatial cognition as sort of a neural circuit or a set of neural mechanisms. And so that's all well and good, but then you have this behavior. And this behavioral output is related to that processing and how much information, specifically how much regulation, goes into those movements. So this again is kind of thinking about simplistic movements that result from maybe like a small agent, like a small connectome or a vehicle. And so we have these different types of movements going from random and Brownian motion to levy flights, where you kind of move around randomly but visit specific locations. And then you have ballistic movements, which are just kind of like sort of moving in a certain direction and then moving again in a certain direction, which is moving directionally but uncontrolled. And then 4, you have first order prediction and dynamics. So once you enter prediction in the mix, you bring in feedback, you bring in this sort of these dynamics that are smoother. You can explore areas that are sort of controlled in a curved manner. And then finally 5, you have higher order prediction and dynamics, where you can smooth out your curve and you can actually make these very intentional, very controlled movements. Maybe you're trying to reach a prey or something like that. And there are a lot of different behaviors in insects, which are very interesting in terms of prey capture, bats as well, where they kind of move towards their prey, they kind of move past the prey and then back towards the prey. And that's all based on their ability to get sensory information to update their spatial map and then capture the prey successfully. So this sort of typology of behaviors is something we're kind of working on as well, and we want to fit this into this model of spatial intelligence. So next is the path integration and taxis view, and I say photo, chemo, thermo, thigmo, because those are the different types of taxis that we want to talk about here. So phototaxis are light signals, chemotaxis are chemical signals, thermotaxis are thermal signals, and thigmotaxis are touch signals. And taxis refers to when you have an environmental signal in that form, and then it forms a gradient in the environment, and the agent or the organism will follow that gradient from where they are to the highest concentration of that gradient. So the taxic movements are organized not by some great regulatory mechanism, but just kind of following the gradient at the next highest concentration. So this kind of relates to path integration. So I'm going to talk a little bit about path integration, which is sort of wayfinding or homing, if you are familiar with like ants or something like that, or insects, where they use path integration quite extensively. And it's kind of remembering space and remembering sort of the map of the space around you, and then kind of going from a home location, knowing how space is transformed, and then moving back to the original home location. So you explore space a while, and then you can always find your way back home. And so that's path integration. It has an interesting relationship with taxic behavior, and we'll get into what that is. So we have these 2 papers, possible uses of path integration in animal navigation, and then principles of insect path integration. And so this is path integration in ants. And so you can see it's a very complex set of behaviors. It almost looks like our second phase of behaviors, the levy flights, where they kind of move in a certain direction, but they make these leaps. And this is kind of, what happens in ants, where they leave home, they forage in the environment, and then they have to return to the nest. And so they're using their ability, they're using pheromones, they're using their ability to navigate and remember space and remember how it transforms when they turn their bodies and then make it back home. So path integration is the basis for cognitive mapping. Path integration will happen in other organisms as well, including mammals. So this isn't just a thing that you find in insects, but insects make great use of this. So this is the basis for cognitive mapping. There's a limited use of landmarks in resetting processes, so they use landmarks, but not exclusively. They also reset their calibration tool, so usually they measure footfall or something like that, and they use that as a heuristic. They also have reset their footfall information sometimes. So there are all sorts of mechanisms involved in this. So the information needed to compute a trajectory involves a limited approximation of distance when the target is close. But when the target is farther away, you need to have a little bit different strategy. Sometimes you can use the direction to goal, which is fixing and resetting your position, and then matching behavior, or having a coordinate system on top of a map and having that coordinate system be the guide to getting back to home base. And so there are these different ways in which information can compute a trajectory and perform this path integration. Now, there's some very interesting connections with taxis because, like with path integration, taxis are cases where the organism goes out, explores their environment. Maybe they're foraging, or maybe they're doing something else, trying to find some delicious meal of some chemicals or something like that, and then return home or return to some other place. And so instead of sort of these other mechanisms that we think about, we actually have these gradients that one can follow. You follow the gradient to its source, and you then find your reward. So there's this very clear signal, environmental signal, of where to go. But nevertheless, you have to, it's not a straight line, it's not a straight shot to the source. There's a lot of exploration, and there are mechanisms you need to use to compare two different places where you visited so you know where the greater concentration of this thing is. So we can actually build this interesting table, which is to generate a sort of a contingency table for any one position as you're tracking or whatever it is towards the source. So there are three different behaviors. You can go towards the source, which is positive. You can partially go towards the source, which is angular, or you can be negative and move away from the source. And so this is all based on local information, comparing two different places where you visited and moving in that direction. And as you noticed with path integration, the path is wavy and it's curvy, and it isn't a straight line. And then we can use different types of sensory information. So we can use light, photo, we can use chemistry, chemo, we can use touch, thigmo, temperature, thermo, hardness, duro, and electricity or electro. So these are all known taxis. And sometimes you can use more than one taxis. You could use phototaxis and chemotaxis, or phototaxis and thermotaxis, and integrate them in a way that helps you to improve your performance. So for each of these, then you can say, if you're at a phototaxis, you can move towards the phototaxis or towards the thermotaxis, or you might move towards the phototaxis and partially towards the thermotaxis. And this is all based on local information. So over time, it'll average out to this path towards the source. But it does produce some interesting dynamics, especially when you have multiple modalities. And so that relates to path integration, of course, because the source might often be some sort of, food source, and then being able to return home from there is also a function of sort of following the outbound signal, or maybe following sort of the decreasing gradient. These are mechanisms that don't involve the type of hippocampal spatial cognition we talked about earlier, but is nonetheless part of the spatial intelligence. We can also look at agents, and we did this interesting work with agents where we were doing this sort of dendritic computing with agents. So we had these Bradenberg vehicles with antennas, and the antennas have these different sensory sensors on them, and they take in environmental information in the spatial array. So you have sort of this beamform of spatial information going from left to right, and it comes into this antenna or this array, and then it encounters these transformation points before it goes in the nervous system. So the way dendritic computing works is you have sort of the analogy of dendrites in the brain, where you have these tree-like structures where environmental information hits the tree-like structure, and all of these sort of parts of the array interact with one another or maybe the ones to the left take in more information than the ones to the right, given the sort of the direction of the source of the information, and then that influences how it's processed by maybe like a logic gate or some sort of transformational process before it gets into the nervous system. And so this is an interesting sort of paradigm because you can do a lot of interesting things with spatial processing. You can have environmental information that is directional. It could be far to your left or far to your right, or it could be right in front of you. And that information is processed differently, even though it's the same source. So if you think of taxis, of course, your taxis are going to be different. Your information is going to be maybe refined, or maybe it'll be obscured by this kind of paradigm. But it's going to influence how things happen. And of course, this is sort of how, if we care about embodiment, we're going to have this sort of array of sensors on the outside of the organism. It could be a visual system, it could be other sensors. And so we need to consider how those are processed spatially and how that influences behavior. This is another example of sort of linking phenotype to this sort of processing where the surface perimeter is very curved, and you have these dendritic trees, or these arrays that sort of conform to the shape of the body of the agent. You have what we call sensory beamforming. This is an idea from engineering where you take the different orientations of sensory information and process it based on its spatial location or its spatial orientation to the sensor and then to the organism as a whole. So this is very different from the way we kind of usually think about spatial processing as sort of like this undifferentiated signal that gets processed in the brain. This is actually it's processed in the placement of the sensors on the body. We also have sensory modality specific gradients. This is from another paper I published. So in this case, each sensory modality forms an input vector. And this doesn't necessarily need to be an embodied agent, but this is like where we have different sensory modalities and the agent is sensing the modalities, and the modalities form a gradient in the environment. So each sensory modality forms an input vector to the agent. A directed vector describes the direction of exploration along the sensory gradient for each vector. So each of these vectors describes a certain modality. It describes a direction in which it's located and the direction of exploration that's expected. And so you can have these different vectors that are aligned or they're misaligned. And so sometimes visual information is in a different place in the environment than auditory information or touch information. Those all have to get integrated. Either, sometimes you'll have an angular displacement, sometimes they're all coming from the same direction, and so that's easy to determine the sort of the direction of behavior, expected behavior. And so this fits into this model of open loop intelligence augmentation, where we can have this model of intelligence as an open loop amplifier. This is something like what W.R. Ashby predicted in his cybernetics work, but instead of having this sort of model of the environment and sort of like an intelligence or an internal model, we have an intelligent agent with a world model, and then we have this formulation of intelligence, which is sort of the sensory information that's incoming, feed forward behavior, the behavior that's generated, and then a selection mechanism on that sensory information. So sensory processing is incoming sensory information with selection. That determines your initial sort of intelligence, and then there's some model of the world that you refer to refine that. And so thinking about spatial navigation in a mathematical framework, we're interested in spatial navigation as this continuous set of dynamics. So we can use a Lagrangian to describe this. We can draw this from the equations of motion. So we're interested in spatial navigation, not necessarily as an agent that's visiting a number of locations and looking at the pretty squiggly lines that it generates, but we really want to know what the equations of motion are, because the equations of motion can tell us something about the behavior of the agent and in turn what they're doing to process information to produce this trajectory. So this is a kinetic description of navigation where the segments of motion or output of place in grid cell representations, for example, or other types of spatial representations are put into the environment as behavior. And this is an active process. So as representation changes, so too do the equations of motion. This is a sketch of maybe what this looks like in terms of the taxis. So we have an agent, a Bradenburg vehicle. It's integrating taxis from multiple sensory sources and produces this Lagrangian description, or this trajectory that goes through space. You start near sort of a thermal gradient, so you get these thermal taxis, and then you leave the thermal gradient and you go to the chemical gradient, and you venture through the chemical gradient. You come back out to the pH gradient, and then you venture through that. You come back to the thigmotaxic gradient, and then out. So you explore these different gradients as you encounter them. Sometimes the gradients overlap, but it basically describes sort of the spatial behavior in terms of these guiding cues, and then the internal mechanisms. That's something in this model, it's a black box, but we can think back to some of the potential mechanisms that we might use to produce this behavior. So I want to go back to sort of the internal aspect of intelligence and talk about the neuroethological view, neuroethology being sort of the study of the brain in naturalistic contexts. So again, we revisit this idea of spatial congruence. We saw that back a few slides back that we had those sensory vectors that maybe they were aligned and maybe there was some displacement. In this case, we can see kind of what that looks like. They train these cats to respond to signals that are both congruent, meaning that a visual signal and an auditory signal are coming from the same place, and signals that are incongruent. So the auditory signal and the visual system, or the visual signal, are coming from different places. And then the food reward is where you get the food right. So, if the signal is coming from the same place, the cat goes to that food tray, gets the reward. If the signal is coming from two different places, the cat has to sort of figure out where the food reward is. Is it the auditory signal or the visual signal? And they have to disambiguate the two signals to get the food reward. So discongruous sensory cues reveal spatial biases. Not all locations or orientations are equivalent, so it has to sort of sort through, even though it has all these orientations and rotations, spatial rotations, and spatial location information stored in its internal model of the environment, it still has to disambiguate a lot of information to make decisions, to make the right decisions without error. We have this model of coincidence detection, spatial coincidence detection, is rooted in the superior colliculus. You can see a model of superior colliculus at the lower right. And so these experiments are very famous for revealing yet another place where spatial processing happens. Related to this is the chemoaffinity hypothesis. This is another famous experiment where it's more about spatial framing. So the idea is that you have, in Sperry's experiments on this, he took a frog and he cut the eye out and he rotated it 180 degrees. So when the eye is in its normal position, the frog can shoot out its tongue at like a fly and capture it pretty quickly and do this pretty accurately. But when he moves the eye 180 degrees, he rotates it in its socket, the tongue goes in the opposite direction of the fly, almost consistently. And so the idea there is that you have this spatial frame that's based on the eye and the way it develops. That spatial frame is put into place. And then when you rotate the eye, you're basically shifting that spatial frame 180 degrees off. The spatial representation of the optic tectum is established in development, and it stays like that throughout adulthood. When you shift the sensory array, the corresponding reference frame is shifted. So again, we have these two cases of spatial incongruity, one where you have two senses that are decoupled, the other where you decouple the sensory array from the target in the environment. And so that leads us to this neurosimulated Morris water maze. And so this is interesting because you have visual and auditory sources, and they're decoupled from each other. Not only are they decoupled, but you have multiple sources around the arena. The idea is these vehicles will travel and try to find the corresponding visual and auditory sources. So there's kind of like this spatial gradient of visual information and auditory information. And in some places it's mixing, in some places it's independent. And so these vehicles are kind of exploring their space trying to fix on some source and exploring its space. And so this is kind of a continuous maze. If we think back to the mazes that we used for, hippocampal learning, this is a continuous maze, and this is something that will, maybe tell us something about how we can process incongruous spatial information in very simple nervous systems. And one more detail about this is that sometimes these sensors are all located sort of in the same direction. You have on this vehicle here, you have 3 sensors all pointed forward, but on this vehicle, you have sensors that are pointed in all different directions. You basically mutated them so that they're on different sides of the body. And so that affects processing. Sometimes that leads to a periodic orbit. Sometimes it leads to something else. And we don't know. We haven't run these experiments, but this is just kind of a thought experiment where, we're kind of thinking about varying the sensor location, varying the sensory source location, and then having this naturalistic behavior where we can observe gradient following and output behaviors that are spatially interesting. So if we have the spatial incoherent framing plus spatially incongruent multisensory information, it can lead to really interesting dynamics. So then the next 2 papers are the brainstem integrator for self-location and memory and positional homeostasis in zebrafish, and topographic maps of visual space in the human cerebellum. So this starts with a quote from this Van Es paper from 2019. That's the second one, topographic maps of visual space in the human cerebellum. And this is as follows. Retinotopic mapping under fixation precludes a distinction between purely retinotopic or eye-centered, craniotopic or head-centered, spatiotopic or world-centered, and other spatial encoding schemes. This is in line with what we've been talking about. We've been talking about different types of sensory-centered processing, head-centered processing, head direction cells, etc. And then this allocentric view of the world where things are spatiotopical. We have this retinotopic mapping that in humans, at least, or in mammals, precludes these distinctions. And so this actually points to the cerebellum as another site of spatial processing. The cerebellum encodes multiple polar coordinate maps of visual information that provides information to the effectors. So now we're not talking about the sensors, we're talking about the effectors. So now this is the outbound information, and the cerebellum encodes these maps for that outbound information to be transformed. Okay, so now we're dealing with embodiment. We're dealing with the consequences of embodiment. The cerebellum is known for producing smooth movements. It's involved in motor control. It's involved in smoothing out motor control. And having these predictive models is very unique architecture, allows you to do that. And O'Keefe and Nadal, of course, also had something to say about embodiment, which is that the movement of an observer's body changes position, but does not alter the position of objects. This is distinct from relative position. The body changes position, of course, as you move. It doesn't alter the position of objects, though, because you have sort of a framework for thinking about different transformational views on objects. So if you're looking at an object from the front or from the back or from the side, you can put that together and know that that's the same object. This is a graphical abstract from this Yang paper in Cell. This is a zebrafish. It shows that the cerebellum and the inferior olive are involved in this location error signal, and then it's also involved in determining velocity, and also then the zebrafish is moving with a change in position. So it's locating the error signal, it's correcting the error signal, and generating a certain velocity that changes the position in an adaptive manner. And we can think of this as a control system where we have this input that leads to swimming behavior. So we have the neural coding, the behavior, and these brain-wide circuits that control this centered on the cerebellum, the inferior olive. And so we can think about this in terms of a map. So we have our cerebellar map. It's taking in information from different parts of this organism. So we have a lot of things in the head that are mapped to things in the hippocampus, and then those are produce outputs for those things. So we have output behaviors being mapped from the hippocampus to output behaviors. And this is very much like sort of a homunculus map that you see in the neocortex. And so, we can actually replicate this in a vehicle. We can have this little area here where it maps sensors from different places to these maps, and then an output to these effectors. So you have inputs, and they get mapped to different cell populations, which have sort of a mapping to a specific effector output. So in this case, in vehicles or wheels, and you have these sensors. They could be light sensors or vibration sensors bringing in information. And so, we can kind of classify our different inputs and our different outputs. And this is kind of what you see with the topographic distribution of output neurons in the cerebellar nuclei and cortex to somatotopic maps in primary motor cortex. So this just shows for different effectors like hind limb or proximal forelimb or distal forelimb, there's a different distribution of cells for each of those types of output in the cerebellum. And so looking at this more closely, you can start to build a network from these different cell types. It adds a functional layer to these cell types in vehicles, and it gives us this ability to map things outward as well as from the sensory system inward. There's this core sensor effector mapping where you get spatial biases in these interneuronal networks. You get information exchange among spatially sensitive interneurons. And then peripheral processing networks, which are shown in the different colors here, are specialized subnetworks for timing, differentiating, and integration of activity. So what we're trying to do is build a very small nervous system that replicates some of these findings. So next I'd like to talk about these two papers. The first is omnidirectional sensory and motor volumes in electric fish, and then spatial planning with long visual range benefits, escape from visual predators in complex naturalistic environments. So this first example shows the weakly electric fish. And weakly electric fish are interesting because they send out this electrical signal around their body. And these are to sense prey in an environment where they can't really see anything. It's a very murky aquatic environment. And this is what they call active sensation. So the electrical signal that's sent around their body from the electric organ near the head to the tail where there's a receptor is this electrical field, this volume. And so in the first paper that I talked about, they've reconstructed these sensory volumes around the fish at different orientations. And then these dots involve sort of prey that are in the electrical field so that the fish can sense the location of the prey so it can move towards those prey and capture them. So you can see that the prey are spatially located, spatially segregated within this volume. And then the fish have to make a movement towards that. And of course, there are different perspectives on this volume. So if you look at the side view or the back view, you can see that there's a different distribution of things. And so one of the other things that they explored was this habit-based versus plan-based action selection. So once the electric fish senses that they're prey within the volume, they can go and capture the prey. But they have these two strategies called habit-based and plan-based action selection. Basically, they did these simulations where you could look at, what was involved in this. And I'll just tell you right now that habit-based is not predictive and plan-based is more predictive. And so habit is based on a model-free approach. So habit is where behaviors generated by state action values are not related to immediate outcomes. They're not adaptive. They're just state action values, and there's no real prediction there. And then planning is a model-based approach where behaviors are based on action outcome knowledge structure. So a knowledge structure that helps prediction. Future states and expected outcomes are modeled, and this is very adaptive. So, kind of what future states to expect, what outcomes to expect, and it's much more predictive than the habit model, which is kind of just output of behavior. And so these are interesting because we talked about in last year's talk by our group about the role of habit in planning in sort of agents and thinking about cybernetics. It's what they call goal-directed behavior. And so this is an important distinction that is also important in the spatial intelligence paradigm. Finally, we want to talk about the view from embodiment in GSLAM. GSLAM stands for Generalized Simultaneous Localization and Mapping. So again, we've talked about embodiment. We haven't talked about GSLAM. And so we'll talk about two GSLAM papers. One is by Adam Safran and colleagues, Generalized Simultaneous Localization and Mapping, GSLAM, as unification framework for natural and artificial intelligences. And then this other review of visual SLAM for robotics, evolution, properties, and future applications. So in these papers, basically, they think about spatial processing as the sort of control system. You have a control mechanism, U sub t, discrete time steps t sub I, and then sensor observations O sub t. So you have these three components. Then it leads to an agent state X sub t, environmental map M sub t, and that leads to spatial representation and behavior. So it kind of looks almost like a reinforcement learning model with input from previous states, the environment, and then it gives you the spatiotemporal representation. But of course, there is this aspect of the classic Sidney Harris cartoon where he's very vague in step 2, and he says, then a miracle occurs. And so we don't necessarily know what the filler is in this between agent state, the environmental map, and then defining those, and then what leads to spatial representation and spatiotemporal representation and behavior. It's a hazy box. Maybe you can see someone inside of it, and I think the things I've talked about in this talk, at least from the biological standpoint, can help. But we could also think about Safran's idea of latent SLAM. And this is a basis for map updating. So we're not totally lost here. In SLAM, we can use these kind of latent spaces to help us simulate spatial behavior and then things beyond that. But latent SLAM is an evaluation between currently represented and novel experiences. There's what they call view pose matching based on place cell and grid cells. So they use the place cell grid cell model. So when current observations match information, then loop closure in the representation happens, and that's confirmation that that observation is in the representation in the latent space. When no node can match existing information in the representation, new nodes are inserted. That's this graph relaxation. So basically what happens is you have this latent space that grows with experience, and so you either confirm that you have this experience of the latent spaces, or this structure of the latent space is either confirmed or disconfirmed with environmental information, and then when it's disconfirmed, you add nodes to represent that. So next time when you encounter something similar, you have that representation in place. And so a couple words about the loop closure algorithm. This is from Introduction to Loop Closure Detection in SLAM. This is where you have a form of error accumulation that occurs similar to path integration in animals. And so in path integration, as well as for this form of error integration, you count the steps along a path, and when a landmark recurs, the map is updated to reflect that information. And so if we state this kind of in a pithy way, we can say, keep wandering until you find a loop closure. Wait a minute, I've been here before. So you wander down the beach, and you keep going, and you find a loop closure until you kind of go all the way down the beach and back to another point that you've been to before. So there are connections between this kind of latent space approach and a cybernetic system. So latent SLAM attempts to recapitulate a neuroanatomically interesting latent space model. Latent space allows us to compress a full state space where we don't need full knowledge of every state. In control theory, state space models serve as a path to dynamical systems, like we talked about with the Lagrangian and all this about trajectories. In a cybernetic formulation, however, we can build upon latent SLAM by using a state space, a growing network, and causal relations. So we can actually formulate a solution to this in the form of a cybernetic model. And this is something I think we also talked about last year. We'll get to this in a minute. But this is an example of graph relaxation algorithms. So this improves your Eulerian path estimates for your network structure by checking if a shorter path can be found through alternative edges. In this case, adding edges. So you explore this network, you see what the path lengths are for each traversal of the network, and if If it can be found, great. If not, it adds a node and you have this shorter path. But this is kind of the way you can do this in a cybernetic context. These are these complex referential or quasi-causal relations for different control outcomes. This is from last year's Symposium, where we proposed this cybernetic model of feedback loops that produce not just the past, present, and future, but also alternative futures and complex outputs associated with all of them. So you can model a feedback loop as sort of going back to the future, basically, as you have in a first order closed loop feedback context, and then it produces a complex output with a lot of different causal consequences and with a lot of sort of causal implications. But then you also have these alternative futures that can be generated from the same system. Another way to think about this in space is you have these Teletubbies that are, there's one in the present, maybe they were here in the past, here in the present, and then you have these alternative futures, maybe a future state over here and an alternative future state over here. And there's a spatial location associated with each of them. This cybernetic model recalls Douglas Hofstadter's I Am a Strange Loop, where in chapter 4 he talks about loops, goals, and loopholes, and he talks about how self-applied operations can produce these circularities that are very interesting, not only from a causal standpoint, but from a spatial standpoint as well. And so we can also think about ergodicity and how that can also contribute to this sort of spatial learning. So ergodicity is this idea that if you visit enough locations in a space, you can produce a mean path that describes every location in that space. So the mean value is something that gives you full spatial knowledge of that domain. And so this is a paper where they're using a robot to learn all the possible states of a movement path. The mean path is described by robotic systems. This mean path is optimal. And this can actually help assist people with disabilities, people who can't move optimally in space. If you have a robot that can sort of explore all the spatial locations in maybe like a movement space, like a workspace, or in a room or something like that, then you can assist people when they make mistakes and help them cover up their mistakes through knowing sort of what the mean value should be at any location. So again, our graphic here in our pithy statement, if you walk down the beach enough times, you can predict a path that matches the lap of the waves from a mean value. So you can actually track the path of the incoming tide and walk right along the water's edge if you've walked along this beach enough. This brings us to this idea of an ergodic ensemble, which is where if you sample a system enough times, any given ensemble is equivalent to the time average. That's sort of the formal definition. Learning increases information, but an internal representation may provide ergodicity. So an example of this is for native language speakers, a language tends to be ergodic, versus English as a second language speakers, where the language tends to be non-ergodic. And this is from a study where they looked at native speakers and English as a second language speakers. You have to build this strong internal representation characterized by sort of native language ability. When described in Bayesian versus statistical mechanics terms, so ergodicity is usually described in terms of statistical mechanics. Individual cases are not equivalent to generalized maps. And so this is something interesting about this, that we can use this kind of ergodic example. We can use ergodicity to describe spatial navigation, but we also have to take individual experience into account. Sometimes spatial navigation for one person is not ergodic, but for a population it is. So what have we learned? The first thing is that a representational system that is anatomically explicit, like such as the hippocampus, and evolutionarily conserved, so the hippocampus in vertebrates, provides the hardware for quick, efficient spatial learning. So the longer you have that structure around evolutionarily, the more refined it can be, the more ergodic if you acquire something in development, then it's ergodic and you have the hardware to carry that out. If you have to use sort of an expedient neural architecture for this to learn it, or it's not something that is typically learned from development, then not going to have as efficient a spatial learning. Number two, optic flow mediates spatial perception in terms of both the body, egocentric, and environment, allocentric. And so we have these different frameworks for the body and for the environment. Optic flow mediates spatial perception in that way. Viewpoint representations or frames of reference also depend on sensory specializations established in development. So technically the sensory specializations are established in evolution, but you can have these things play themselves out in development, and they influence the viewpoint representations across species and how, for different sensory systems, for different neural architectures, they actually mediate spatial learning. Number three, path integration produces dynamical trajectory with complex parts. So this is, again, path integration, another form of spatial intelligence, where you get this dynamical trajectory, where you can decompose the parts of the trajectory, and it tells you something about periodic spatial intelligence, or at least the behavior of spatial cognition. And so path integration results from sensory and internal information, as well as processes of regulation and prediction. Number four, a body can be characterized by a sensory manifold, while sensory motor integration acts as an immediate source of spatial bias. So again, we have this, the body plays a role, and then sensory integration also plays a role. And so sometimes our body is getting visual information from one location and auditory information from another. Sometimes our bodies can act to bring those together, sometimes not. And so this is an immediate source of spatial bias. Action selection operates on the integration of immediate or a longer term information. So we can have a simple model of immediate sensory information, like in the habit case in the electric fish simulations, or a longer term model of planning, or sometimes we can use a world model like we talked about in the intelligence augmentation example, where you have this model of the world that you draw from. So it's a longer term context. Number five, unified models such as GSLAM serve as a framework for embodied information processing. This unifies the role of hippocampal path integration, circuit, optic flow, and the spatial bias of sensory reception. So GSLAM really unifies a lot of these mechanisms into one framework. And then six, which is an additional thing, and this is kind of more of an insight, enough free or random navigation can produce a good enough cognitive map by relying on an ergodic ensemble of spatial information. So if we just let our simulation run forever, our agent explore forever, it can produce a good enough cognitive map. It doesn't necessarily need a lot of architecture, but the architecture deals with a lot of these other aspects of structure that random or free navigation may not solve. And so again, with the neuro, we're dealing with potential brain centers, connectivity, mechanisms for spatial representations, and cognition across animals. The simulate, we're using boxes and arrows or cybernetic type systems, spatial distributions of sensory information, mathematical functions that describe behavioral output, simple biocybernetic agents, and models of sensory processing from the literature. So in future directions, we need more complex models, more exploration of causality in systems context, expansive and reconfigurable networks, and of course, more synthesis of all these ideas. Thank you for your attention.